Okay, it's 2 after, and we have a lot to cover today. So let's get started. First, hi everyone, and welcome. Thanks so much for being here to learn more about this useful resource we've worked together to create. We are going to present a checklist for researchers today that was designed to help strengthen and improve data submissions to generalist repositories, especially those that provide persistent identifiers and clear licensing for reuse. Next slide, please. We are your hosts. I am Jess Herzog, and with me today is Crystal Steltenpole. And we're your host for today, and we're also supported by other GREI team members in the background who will help guide us through our program for today. Next slide, please. We are really excited to welcome our guest speakers today, John, Wendy, and Troy. They're here to share their experiences with submitting and publishing data in generalist repositories, along with their key takeaways and benefits they've experienced in their data sharing journey. They are without a doubt the main attraction and will be the most impactful voices in today's conversation. So we are very lucky to have them sharing their insights with us, and really thank you for joining us today. Next slide, please. This is our agenda for today. Essentially, like I said, we will do a brief welcome, an icebreaker. I'll introduce you to GREI and then hand it over to Crystal to walk you through the checklist. And we'll also hear from our researchers. We'll review resources and a Q&A at the end. So, here's the icebreaker. So we're going to pause for a moment here to get to know you a little bit better. If you have attended the previous session in this three-part series, you may have seen these opening questions before. So, and we would appreciate if you would respond again. We have some new questions for you later in the session, but I'll hand it over to Lisa to guide us through these. There is a QR code and a link. Is there a link there to join the Slido?
Yes, there's a link in the chat.
Oh, thanks. Okay, link in the chat. Which city are you joining us from? Where's Norman? Boston, Fort Collins, Tampa, Mexico, Cincinnati. Wonderful. Buffalo, Boston, Roanoke, Princeton, Seattle, Chicago, Charleston. Awesome. In here with this question, we're just attempting to capture who's with us today. So please let us know and share your role. Lots of librarians, great. And here we're capturing the GREI repositories, which I will introduce you to, that you've interacted with before. And you can select more than one response here. And these are great resources you previously have encountered or used. Great. The 7 repositories that are part of the GREI community have worked hard to create these for our researchers. I believe we've had, yeah, please keep your responses coming in if you haven't responded yet. And I know that the comparison chart was recently updated, and we will soon have a new version of the flow chart coming out soon. So look for that. Thank you. So I wanted to talk just a little bit about GREI for those of you who might be less familiar. GREI stands for Generalist Repository Ecosystem Initiative, and it's an NIH-funded initiative to improve data sharing, discoverability, and reuse for NIH-funded research. It connects the 7 generalist repositories listed below: Dataverse, Dryad, Figshare, Mendeley Data, OSF, Vivly, and Zenodo. We're aligned in our approach to fair data sharing. We follow best practices to support consistent standards, high quality metadata, and training around FAIR principles, which FAIR stands for findable, accessible, interoperable, and reusable. And these principles are designed to make research easier for both people and machines to discover, understand, and use. The NIH encourages researchers to share their data in domain-specific repositories whenever possible, but not every data set fits into one of those specialized spaces, and that's where these generalist repositories come in. They provide researchers with a reliable place to share and store data that doesn't require um, quite a match to a specific domain. You see there, GREI's mission is twofold. Um, and while you're reading through those, I do want to share some key activities that we collaborate on and work together on. One of those is developing consistent standards for metadata, data citation, and repository metrics across platforms. We also work to conduct training and outreach on data management and the importance of data sharing, particularly with NIH-funded data sets. And that's part of why we're here today. And we work to improve discovery, making it easier, as I mentioned, to find, cite, and reuse data across multiple disparate generalist repositories. Next slide, please. So I'm going to hand it over to Crystal now, who will walk you through the ins and outs of the GREI checklist for researchers that we've developed.
Hi, everyone. So yes, let's talk about this checklist, and hopefully we can get a link to it in the chat as well. So let's take a look at what the checklist has in it. So if we can go to the next slide. So this checklist is really, really helpful both for people who are managing their own research projects and also for people who are assisting those who may be managing their own research projects. It is a checklist of some of the most basic things that you want to make sure that you're doing as you're moving through your project and how to manage and share your data throughout that. So we've split it into kind of 4 broad categories. The first is like planning to submit your data. So this is where you're actually planning your study out and you're saying, okay, what am I going to need now before I've done anything? that I'm going to plan for so that when I'm getting ready to submit, I already have all of that stuff connected. So this can include stuff that you might do as part of a data management and sharing plan, for instance. The second part is preparing to submit your data. So this is where I've collected my data, I have everything together. I think I want to start getting it together for a repository. How am I going to pull? Just make sure that everything's organized so that when I actually submit, it's nice and easy. Submitting your data for publication, that's publication to the repository. This is going to be some of the stuff that you're going to want to check through as you're actually going through the submission process. And then after you've submitted your data, it's on the repository, it's got a DOI, you can link to it. Here are some things that you can do. So we're going to go through each of those sections. I'm going to tell you what's in each of them, and also talk about some of the resources that are in each of those sections. So on each one of these pages, there's going to be like a brief description of what each of the major tasks are in that phase, and some of the resources that are available. So, and then at the end of the document, at this checklist, there's also some other additional resources that may be helpful for you or whoever it is that you're assisting with as they're working on their project. So let's go ahead and dive in. So in this first section where you're planning to submit your data, right, we want effective research planning and data management because that helps to make data sharing faster and easier. It's helpful both, right, for people who might be looking at your data. It's also really helpful for you because you will know where everything is and you will know how you're going to use your data. So that's going to be helpful. So some of the first things that we recommend that people do is to really start to think about if your research is funded, what are those funder requirements? Some funders don't have requirements, but some do. So knowing whether your funder has requirements and if so, what those requirements are going to be really helpful. So you may actually have to reach out directly to your funder to find out. They may have guidance that's already available and easy to find. And so making sure that that's there is going to be helpful. You're also going to want to start to think about any institutional requirements. And so this may require reaching out to a couple of different offices or checking out their website if they have guidance available. So this could be your sponsored research office. It could be any of your data libraries. It could be a data management services team. It could be your IRB or your human subjects committee. These all have different requirements. If you're going to share data, this is what it looks like. So just kind of compiling like what is actually expected of me as I'm going through this. And then after that, you're going to want to really start to think about any embargo requirements that you might have. So you may want to delay the publication of your research data for one reason or another. So ensuring that you're following any kind of requirements that have that, and that you're thinking about what that timeline might look like and reasoning for that timeline. One resource that we do also talk to folks about is using a data curation primer. So what these are documents that help to delineate specific expectations or guidelines for specific types of data or specific types of research or research that occurs within a particular field. So the Data Curation Network has a wide range of primers that detail what these specific sorts of expectations are. These are also really helpful to use because they take some of the I don't want to say they take some of the thinking out of it, but they take some of the uncertainty out of it that comes with if you're having to come up with all of the things that you need to collect. That can be really overwhelming, especially if you're doing it for the first time. These primers are created by experts within the field, within that methodology, who are used to data sharing. And so they have some really good guidance there for pulling all of that together. And so that can be really, really helpful as somebody, especially if it's your first time sharing data, to go, okay, so here are the things that I'm going to need, and pulling all of that together. And that also can be really helpful for identifying what metadata you're going to need. So metadata is basically descriptive information about your data. It's like the simplest way to explain it. So some things that you might want to think about are, okay, if I was searching for this data set, what would I want to know about it in order to be able to use it? So this could be things like who collected the data, when it was collected, where it was collected. It could be the variables that are involved. It could be code books, any kind of standardized information that really helps to describe what this data set is, what it is for, how it can be used, what format it's in. And that's helpful, again, both for you, but also for future researchers. And so again, there's some guidance that we link to that helps to kind of get the gears going in planning all of this out. And then something that's also, I think, really, really important is actually setting up file naming conventions. This is particularly useful if you're working, if you're collaborating with other people. So making sure that everybody's using the same file naming conventions, that we're all calling the same, similar things, similarly. Is really helpful both for finding and also for things like versioning. So knowing which version of a document to open. That's a lot easier when you actually have a file naming convention as opposed to just like a document that's labeled like data set. So having some more descriptive information in your file names can help keep you organized. And making sure, the specific rules don't necessarily super matter. There are some best practices which we do link to, but having a specific rule is less important than just making sure that it's consistent across everyone that's using it and that it makes sense to the team. Um, so, yes, so, and then the next section, um, is also for particular types of data. You may have to consider the sensitivity of that data. So if you're collecting human subjects data, you want to make sure that you have informed consent for primary research and for future data reuse. So ensuring that your participants understand what data sharing means is also really important. Making sure that you have permission to share that data is going to be a key part of that. And some institutional review boards have guidance on that already. If not, actually, LDBase is a data repository for educational research, but it actually has some nice language that I think applies in broader than just education research that you can use on informed consent, both when talking to your IRB and also in the consent forms themselves. And that's actually true for a lot of our resources. Some of our resources are a little bit more subject specific, but we found that the guidance that's in there can be expanded beyond that particular subject area. You may also want to be considering how to de-identify data. This can be more complex for certain types of data, for instance, qualitative data, where simply taking out an identifier might not be enough. So really thinking through how do I de-identify this data and still maintain its utility for different researchers. You might want to start planning that before you actually collect the data. It does make it a lot easier. And again, we have some best practices linked for ways that you can de-identify that data. You may additionally consider something like managed access or controlled access. This is where if it's completely open access, you just kind of upload the file and then anybody can access it. Some data may be sensitive enough where you actually want to ensure, for instance, that it's a researcher that's affiliated with an institution that's downloading it. And so some repositories offer this as a service, some don't. So you want to make sure that you're starting to think, do I need that? Do I want to ensure that only certain kinds of researchers or certain kinds of folks are able to access it? Or is it not so sensitive that I can upload it without that? So really starting to plan for that and to look for resources. We've got again, some resources connected to that. You may also, if you're working with particular Indigenous communities, you may also want to double check things like there may be some specific laws or regulations, also some best practices for working with Indigenous communities that you may want to consider or you may need to consider depending on the nature of the work that you're doing. And then finally, for this section, you may also, if you're working with species data, you may want to double check that you can actually share that data safely and that's not going to harm conservation efforts or expose any vulnerable species or habitats to undue harm. So you want to, we've got some guidance from Dryad there on how to ensure that. And then once you kind of have all of this set up, so you're thinking, okay, so this is the kind of information that I'm going to need to collect. This is how I'm going to have to organize it. Here are some special considerations for my specific kind of data that I'm going to need to take into account. That all helps you to get to where you're budgeting for if you have any data submission and preservation needs. You may need to go to a repository that has specific charges to it, so you want to make sure that you budget that. You may realize, oh, this is going to take some time for me to do certain things with my data, so you want to budget for, your staff, the people that are in your lab to actually do this, both in terms of time and other resources. So really taking that into account and building that into your planning process is going to be really helpful. And like I said, with all of these different specific sections, we do have links to resources. And again, also at the end of the checklist, there's a number of other resources that are available for people who want to dive into a particular thing where you're like, oh, actually, my data, I'm going to really care about this a lot. You can actually dive in and do that. So now you've planned, right? You've gone through these checks. Some of them may not actually be applicable to yours, so you can just say, okay, I can skip over that. And we actually have a couple of questions to get you into the next section. So the first, a few questions we'd like to know from you all. So when do you usually start preparing your data files for submission? So you're getting ready to submit to a repository. When do you actually start planning for that or preparing for that? And also just curious if there's anybody here that's actually used this checklist before. It's still fairly new, so it's okay if you haven't, but just curious. All right, so folks are often preparing to their data files for submission as they're writing the manuscript is pretty common. And that's all right. So this is a new resource for people. That's actually really exciting for me. I'm glad. So that's awesome. All right, so let's dive back in. So now you're either preparing or you're helping somebody else prepare to submit your data to a repository. Um, you're going to want to be thinking about what an appropriate repository is for your data. We've got I saw some folks said that they've used the selection flowchart for the for the generous repositories. So that's a resource that's an option. And you may also, you may especially need to think about this if you're doing controlled access or managed access. So we've got some guidance for that. Once you've selected a repository, or this may even be as you're selecting a repository, you may be thinking about what the guidelines are and what the guidance are. So there are some rules to submitting to some repositories. So both making sure that they can do for you what you would like for them to do, and also that you can meet their guidelines and their rules. So you're kind of having a conversation there. You may actually reach out to those repositories directly. We love to answer questions. You'll also want to start to compile, if you haven't already, what's called a README file, which is basically like a welcome here sort of document. So that's it tells you everything that you need to know about getting started with the data set, who collected it, who to contact, the licensing information, which is also here. If you don't license your data, that means that nobody can use it. So you want to make sure that you tell people how they can actually use the data. So these are all things that you're going to want to start to do. You're also going to want to, if we can go to the next slide, um, to use file formats that are easily used when possible. We know that's not always possible depending on the kind of data you're using. But if you can use an open non-proprietary format, so like text files or CSVs instead of Excel files, using zips instead of RARs, stuff like that, doing all of that can be really helpful in ensuring that other people are actually able to use the data set without having to have complicated or expensive software. Again, it's not always possible. Some data, that's just kind of how it is. But when possible, it's great to do that. And then you're also going to want to start to actually compile all of those relevant materials that are going to help people to be able to interpret and to reuse your data. So this could be unprocessed and processed data. It could be relevant code. It could be other external resources. It could be code or software. It could be publications, stuff like that. You're going to want to start to compile those to make sure that they're available and that people can access them. You've worked really, really hard on this project, or the people that you helped have worked really, really hard on this project. We want to make sure that people can actually see that stuff. And then the next things you're going to want to check. You're going to want to take out any irrelevant or ambiguous content that might confuse future users. Sometimes people are like, oh, maybe I should include all the visualizations for my manuscripts and stuff. That tends to not really be helpful. You can just link to the manuscript and they can see them there. They don't need to actually be a part of the data set. So just really starting to think about like what might confuse people and making sure that that's left out. Some journals also have requirements, so you might also be thinking here about what journals you'd like to submit to. Do they need specific things? They may have some rules about when you can edit, when you can version data. They may also have some rules about when that data needs to be available, stuff like that. And so just making sure that that is an option. Also making sure that you're finalizing author order and corresponding authors, who's actually going to get connected, or who's going to be contacted if folks have questions. And then finally, you're going to want to reconfirm a few things. So if we can go to the next slide. If you're using human subject data, is it de-identified? Have you followed tribal sovereignty? And if it's species data, is it safe to share? And so those are all things that you're going to want to do as you're preparing to actually submit your data to the repository. And then actually submitting your data to the repository, we've got some guidance there as well. So confirming the completeness, making sure that everything is there. Again, if there are journal articles or anything that you need to link to, making sure that those are linked there. Other relevant links that may be helpful, so things like preprints or pre-registrations, journal articles, peer reviews, if you need to share data management and sharing plan, making sure all of that's connected. You might also want to double check that if you need to be in contact with your repository, especially if you're doing something like managed access, or if there's some kind of approval process for submission, making sure that they're not going to your spam email, because they may have a couple of questions for you and you'll want to go back and forth with them. And then also if you are working with a data primer, just making sure that you're double checking that. And then finally, so you've got your data submitted, and it's out there, it's got a GOI, everybody's really excited for you. Some of the things that you can do after the data is published, you can version the data as necessary. If there's something that you need to make changes to, make sure that you outline what those changes are. Include that in your README file and make sure that the data is versioned. You'll want to link your publication and your data. So from your data to any publications, and also in your publications, you'll want to link to the data. That way, if the researcher is coming to your data first, they can also see all the cool papers you've written. They're getting to the paper first, they can actually see your data. So making sure that those two things are linked is going to be really important. And then, of course, there's, promoting your data. So, you can put that on social, you can put those links on social media, add them to your CV, your LinkedIn, all of that. Making sure that people know that it's there. So bringing awareness to your data. And that helps to ensure that you get proper credit and that people can actually see all of your hard work. So we do have a couple of more questions, and then we're actually going to dive straight into the researcher experiences, which is probably going to be what you guys are really excited about here. We've got some really cool projects. So each of the researchers is going to take about 5 minutes to talk about their experiences, sharing to a generalist repository. But in the meantime, let's answer a couple of these questions.
Yeah, and Crystal, in the interest of time, everybody please jump on Slido and answer these, but we don't need to show the
Sure.
results live.
All
We'll
right.
just move right on so we can make sure we have time for questions at the end.
Perfect. Yeah. So let's do that. Let's go ahead and start with Troy. Troy can talk about his experience sharing with a generalist repository.
There we go. Got my audio. Hello, everyone. Welcome from Buffalo, New York, where we are thawing out. And so I wanted to share my experience with Dryad, which I have to tell you was very, very positive. Although I'm a chemistry professor, a lot of the research that I do is interdisciplinary in nature. So I work with people in other fields. In this particular case, I'm working with plant biologists who are actually more genomicists and experts on plants, where I'm more of a chemist. So, this particular case was we collected mass spectrometry data here at Buffalo for some soybean plants, and we wanted to understand the metabolites that were involved in different environmental stressors for soybean plants. Just so everyone knows, I grew up in Indiana. I grew up on a farm that had soybeans, so this is like me, old hat, back to my Hoosier roots. And but I had this collaboration with University of Missouri, and one of the things that this data set did is we wanted to be able to use a technique that people had been developing for complex mixtures in mass spectrometry called the Kendrick mass defect, or KMD principle, as a way to try to visualize and understand how certain metabolites might be connected to each other in a structural format. People have used this a lot in petroleomics, where they're studying the components of petroleum, but it's been used much, much less often for plants. So we wanted to sort of use this as an example for plants. You can go to the next slide. And one of the things I can tell you where this becomes really important, sharing to a generous repository, is when you have multi-institution collaborations, as we had here. But in addition, I think everyone should consider that for some certain training activities, this could be very valuable. And it turns out that our lab here in Buffalo, we have training activities that involve scholars from Zimbabwe, Nigeria, Jamaica. And so I think for our international trainees, giving them exposure to generalist repositories, and the fact that, hey, they can generate the data here, but then they can still have access to it when they go back home is an important point. So next slide. So I do mass spectrometry, and so one of my big areas is in metabolomics. One of the things that we're using metabolomics for now is trying to identify markers for autism in both urine and fecal samples. We actually demonstrated some differences in fecal samples, and now we're trying to see if that also holds when applied to humans. And in our field, one of the things that we really want to know are there quantitative differences between one population or another? So let's just take the case of soybeans. Let's say we want to look at soybean plants that are under normal growth conditions and another one that's under drought growth conditions. So water has been limited in access. And what can we find that's different between them? Well, in order to quantify, we need to have labeling strategies so that we can quantify the key metabolites that we're interested in and be able to say something about the differences. Interestingly, one of the former postdocs at the lab that we work with in Missouri has recently moved to New York and is continuing plant metabolic stress research, but this term with grapevines because we are New York, Western New York. We have a lot of vineyards. Grapes are an important crop here. And looking at similar environmental stress conditions, but also one that won't surprise you here is cold. What's the stressor to cold? So I think a real key from our experience is that when you have multi-institute collaborations, um, especially when at the other institute there are other experts in the area who may not be part of the research team but could benefit from the data, it's really good to have the data available on a generalist website. Next slide. And so, as I mentioned, I prefer generalist repositories because even though I'm a chemist, I work interdisciplinary with people in all kinds of different fields. In particular, for this study, our collaborators were from another institution, and they were quite literally plant biologists. And also, the journal, and I'm gonna do my little tout for metabolites because I am an editor for metabolites as well. But this paper was submitted, through another editor, obviously. But we require it's one of the things that we think is an important thing at metabolites is that the data be available in a public open access format to the world. And we really want to promote especially the utility of this for people in international areas that might not have access to journal articles per se, but we want the data to be available in an open access format. So given all of these factors, I can say that my experience was really quite positive. And Dryad, because like I said, I appreciate that it's a generalist feature is very important for me. But one of the things I haven't yet told you that turned out to be very valuable during the process was the curation team at Dryad. They were so good at providing direct support to me with the data sets to make sure that others could understand and be able to use how to access it. And they asked me to provide some additional directions, which I think turned out to be really good in terms of accessibility. They wanted to make sure that everything was consistent throughout the data sets. And certainly they focused on the quality of that. I really appreciated the one comment that the curator had is that there are ways that we could probably make this more accessible. And could you take a look at an approach to do that? And it turned out it wasn't that difficult to do. We were able to do it. We incorporated it in the submitted data set. And I think the data set is a lot more clear and usable for people because of it. So that was a very good experience. Okay, so if I were to give you a few tips on why you should consider open data sharing, first of all, using open formats ensures the accessibility. Especially internationally, people don't all have the same tools. We talked about tools that might be proprietary versus open access. So I could collect data that's on a proprietary platform, but I can convert it into an accessible format that others can use. And that turned out to be, I think, really important with this data. I definitely like Dryad because of the fact that they do offer really good curation services, and they're very dedicated, and they're also very fast. I think that researchers need to consider how they want this data available for long-term plans, not just the short, okay, I need to get this up on the web and share it, but is it going to actually be available in 2 years, 5 years, 10 years? Think about that. And especially when you're dealing in collaborative situations with people, especially internationally. And I cannot emphasize this enough, thanks to the curator, provide clear usage instructions, because that really, I think, enables people to navigate your data and um, use it in a way that they might not have been able to if you kept it in a relatively inaccessible format. Now, in terms of practices, it's metabolomics is probably one of the most interesting areas where you've seen a huge growth in data sharing. In the past, and it's because one of the major platforms that we use is mass spectrometry. All of our vendors have their own proprietary software. And I would say in the past, we used to rely on this sort of principle of, oh, if you request the data from us, we'll be glad to share the data files and send them to you. Or send them or give you access on a server. But now we can openly share it, including some of these data sets are just enormous in size. And so having it in a public repository actually makes me more confident. I shouldn't say this about my institution, but it makes me more confident than having it stored locally at my institution, because things happen. And there come times of space limitations and such. So I think a public repository for the long run is a better move. The fact that all of us in the field of metabolomics are sharing this data is making really good advances, because what we can do is we can mine these data sets. And I'll tell you one of the things that I've already done with this data set is, okay, I found certain metabolites that have been influenced by environmental conditions in the soybean. So I want to see what other plants might have shown a similar thing, to see if we're hitting on a potential mechanism that is influenced by drought, let's say. So I can actually go through other people's data and start to mine that. I'll tell you what that does. It either increases my confidence that we really got it right, or other times it means maybe this metabolite is not as important as I initially thought it might have been, and this pathway isn't as important in the adaptation. But I think this is going to have huge effects in metabolomics. So our field is going to be using this a lot.
All right, thank you. Wendy, we'll have you next. Folks can please feel free to use the Q&A if you have questions for the researchers, and we'll ask those at the end after all three have been able to present.
Hello. I will apologize ahead of time if I end up coughing because I am recovering from some sort of plague that has burdened our university. So, I have been using OSF for quite some time because of the many, many benefits. But this particular study was quite exciting because we had this huge data set of over 700 participants where we had collected behavioral data and survey data and a lot of demographics. So we had a lot of potential for future collaboration and work. This particular work was published at Cognitive Neuropsychiatry, where we tried to identified early visual processing differences between non-clinical individuals with autism-like traits and schizotypy using the pattern glare test. That is a simple grading test that is associated with cortical activity. So one of the things that we value about OSF is that it has harbored this culture of accountability and also transparency, where the researchers, we have to put a lot of effort into making sure that our data sets and our data are in a format that is accessible and ready for collaboration. Um, but also that, it assures that the research is transparent. This is our team. Luckily, this study was also a collaboration between two separate labs. And I'll talk about that in the next slide. So we have the Hague lab where we actually focused on sensory processing and how it affects health with populations like migraine and autism and schizophrenia, how the brain processes information, individual differences in sensitivity and discomfort, and how that affects sensory pathways and the mechanisms that are underlying these experiences. And we measure these things using behavioral and also neuroimaging. And then we have the memory and brain lab that is focuses on more of the cognitive domain and measures cognitive processes behaviorally, but also using a wide range of neuroimaging. So this this research focused on um, behaviorally measuring visual sensitivity in individuals with a high number of adult autism-like traits and schizophrenia autism-like traits. Schizophrenia and adult autism overlap in their presentation of symptoms, particularly negative symptoms, and sometimes this increases the risk of misdiagnosis. So the pattern glare test being a very simple, it could be printed on paper, uh, accessible test was a good way of measuring one of the ways they do differ, which is in their cortical activation. Schizophrenia tends to be hypo-responsive and autism tends to be hyper-responsive. So using the behavioral pattern glare test, we're able to see if we could capture these differences behaviorally, and we actually did. Schizotypy, which is the non-clinical population with schizophrenia-like traits, exhibited more illusions on the pattern glare test, and which is consistent with cortical hyperexcitability, and there is no relationship with autism. But despite all this, what's really exciting is that we had this data set with over 700 participants, and we had pattern glare test responses that could be used in several ways, more basic and more clinical. We had the schizotypal personality questionnaire that has these non-clinical schizophrenia-like traits. And then we have the autism quotient that had the non-clinical autism traits, as well as demographic information such as age and gender and all these things that may be relevant for future research. So some of the challenges that we have encountered in the past trying to access other people's data, not using OSF, of course, but through other venues, is that sometimes it required a software that we don't have access to or is not funded through the university. There's security concerns. Sometimes it's just not readable and not understandable. So it's and then sometimes you have to request it, which wastes a lot of our time. And then sometimes we get it and we still don't understand it, which is a challenge that I think is quite widespread. With OSF, we're able to overcome all this, of course, as it provides this generalized platform that is organized, publicly available, and easily accessible, and all these things that just foster transparency and reuse. So like as I mentioned, it encourages this clearer documentation and organization of the code and materials. This is particularly important when working with like large data sets, such as these, where you could really have the time to explain exactly what the things are, link to papers, and also the questionnaires and provide this information of like the authors and collaborators who could help facilitate this collaboration. So, like I said previously in the time, this having access to things that are not user friendly has wasted time and has come with security concerns. But by sharing comprehensive data with clear documentation, this research enables future exploration into like neural markers that may be underlying this behavioral differences that we captured, but also more investigation into non-clinical autism and schizophrenia that warrants future attention, and what exactly we're capturing through the illusions and the visual sensitivity. Thank you so much for listening. I'm sorry I sound this way.
No, thank you so much, Wendy. That was great. And last but not least, we'll have John, and then we'll open it up to a couple of questions.
All right, thanks everyone. My name's John Steffen, and I'm an analyst at Northwestern University. And today I'm going to be talking about my experience creating an R package and sharing it on Zenodo. Um, thanks. So I'm assuming a lot of people here are familiar with R, but if you aren't, R is an open source statistical software program that it's popular in healthcare research and statistics. And when researchers want to share functions and code that they've created, they can share their R scripts, of course. But if they have a lot of functions that kind of work together, they'll often want to create what's called an R package. And that's kind of a standardized way to combine code and data that make it easy to install on users' own computers. And these are often stored on usually GitHub, or the R Foundation has their own network that they store R packages called CRAN. Next slide, please. So these R packages can be a combination of R scripts and data. So the package that I'm going to talk about includes R scripts and R markdown files, data, and documentation. Next slide, please. So the PS Harmonize package is an R package that uh, provides functions that assist users with what's called data harmonization. And data harmonization is a process where if you have data sets from different sources that have similar variables, but the actual values are different, you can, harmonization is where you try to find common values between those data sets and convert them, and then they're often pooled and analyzed. So the PS Harmonize package kind of helps with inventorying, keeping track of these decisions, and the data manipulation part. Next slide. I worked on this R package while working on the dementia risk prediction project, which is a harmonization project aimed at taking dementia incidence data from multiple prospective cohorts. And we harmonized the dementia incident data as well as demographic data and clinical data. So we've pooled and harmonized those data sets. And now we're working on we're finishing up a paper where we created a prediction model for dementia. Next slide, please. So I started working on this project a few years ago, and once we had a package that we were happy with locally, we uh, uploaded it, or we shared it on GitHub a few years ago. And we also wanted to write a paper kind of describing how the R package works and kind of the motivation behind why you would want to use it. And that was submitted in a journal called Patterns in 2024. Next slide, please. And part of the FAIR data sharing requirements was to store our code on a data repository. So we also we chose Zenodo to upload our code to. Um, and after that, we wanted to, increase the places where this PS harmonized package was stored. So we submitted it to CRAN and they accepted it last year. So in a few different places now. Um, and one nice thing about Zenodo is I'm guessing some other data repositories do this as well, but Zenodo will allow GitHub integration. So as we continue to update and continue to develop PS Harmonize, we can create releases in GitHub, and this will automatically push to Zenodo. And Zenodo will it has a DOI for the overall package, but also for each release. So it's kind of nice as a software developer. Um, and that is yeah, that's kind of my experience using Zenodo with this R package. I have a few more links for where to find the package and then I have some references for the paper as well. So thank you for letting me share this story.
Thank you, John. We've got a couple of questions on Slido here. As we continue with this initiative, with the GREI initiative, what kinds of tools or resources would make data sharing and or discovery easier for you? It's open text, so you can write whatever you want in there. And then also, like, if when you're thinking about your role at your institution, what would be something that would be really, really helpful that would improve the use of a data repository for you? So this is a chance. This has got 7 repositories that are listening and ready for your feedback. So if you have some thoughts on that, please do join the Slido link that's in the chat or use the QR code and we can go ahead and continue on. There's a couple of additional resources that Jess is going to cover, and then if we'll see where we go from there.
Yep, thank you. So please continue to respond to the Slido if you're able. Here are just a few screen captures with additional resources that GREI provides. You can link to the full suite via that QR code. Next slide, please. And at the very end of the checklist that Crystal reviewed with us today, there's an appendix that includes guidance for README files as well as metadata completeness. So these are great to have on hand as you are working and thinking and planning about data sharing. Next slide, please. More links to user stories similar to the ones we shared today. The GREI comparison chart and repository selection flow chart go together hand in hand. So I think if we're able to share this out, these are links that you can bookmark and reference for future use. Next slide, please. And if you enjoyed this session, please take a moment to register for our third and final session, which will cover strengthening your data management and sharing plans with a practical guide that we've created. And that will be on February 25th. And share your story. These stories that we've shared today directly from our researchers are really impactful. So we want to hear yours. Feel free to submit them to us. You can scan that QR code for more information, and we'd love to highlight your work. And I will say, I think these user stories also get a lot of views online. So that's a wonderful perk for you. Next slide, please. And thank you. Thank you again to our guest speakers today. We really appreciated your attendance. Are there any questions that you wanted to address that were in the Q&A? There was one for Troy there.
Yeah, I did put a
Great.
response in
Thank
there, so
you.
you can see that.
Wonderful, wonderful.
Yeah, thanks everybody. I mean, I know we're at time. There's a
Yeah.
couple things in the Q&A, but if we don't get, if you have to drop, we are going to be publishing kind of a summary of all three sessions to our blog at the, probably early March that will cover the Q&As from these different sessions. Um, yeah, anything else we wanted to touch on urgently today? I don't know. I know Jess and Crystal, you had some questions for our panelists, but we have gone 2 times, so um, we may follow up with those and share some of your answers when we do our summary.
Yeah, that we don't want to take up too many folks' time, but that might be the best option. We can ask our presenters and share that out with the summary. Thank you all for coming. And please do the Slido is there. Please do respond to those. We love feedback. If you end up using the checklist and you have feedback, please reach out to us too. We are always looking to make things easier for people to do. This is really important work, and we want to make it as easy for folks to do it as possible. So thank you for coming, for listening to our presenters. Thanks to our presenters for sharing their experiences. They're all doing really awesome work, and hope to see you around.